Hello friends, today we are in the session of gibberellin where we are going to study about the gibberellin's signaling in plant cells. So, coming to the topic, here we can see a diagram of a cell divided in two parts, which in one part indicating the absence of gibberellin, and in another part there is presence of gibberellin. Let's read the text first, then we will go into the diagram. Okay, so in this diagram you will see DELA protein. Now what is DELA protein? DELA protein serves as a key repressor in GA signaling. In the absence of GA, DELA protein inhibit growth by forming inactive protein complex with the transcription factor. Then second is GA mediated growth promotion. GA promotes plant growth by inducing 26S proteasome dependent degradation of this DELA protein, which is a repressor basically. Now GA receptor interaction. So GA when come to the towards the cell, it will be received by a protein called GID1 protein. So the binding of GA to its receptor GID1 protein facilitates the interaction between GID1 and DELA. So the formation of there will be the formation of GA, GID1 the receptor, DELA the repressor. So the GA GID1 DELA complex, this is the complex, is formed strengthening the association between DELA and the F-box protein component of SCF SLY GID2. Now what is the full form of these things I will tell. After this, the formation of this GA GID1 DELA complex, there will be degradation of the repressor DELA through 26S proteasome pathway. We can call it as ubiquitination. Okay. So there is release of transcription factor. The degradation of DELA protein result in the release of transcription factor. The transcription factor will be free from DELA protein, and it will be able to attach with the promoter region of the gene responsible for the GA signaling basically. So transcription factor once released activate the expression of GA responsive genes, and ubiquitinated DELA protein undergo degradation via 26S proteasome pathway. So here GID1 is gibberellin insensitive dwarf 1 and GID2 is gibberellin insensitive dwarf 2. Then SKP1 Qlin F box is the SCF protein, and SLY1 is sleepy 1. So these are the full form of the short names represented here. Now coming to the diagram, we saw this part is dealing with the absence of GA, and this part is indicating the presence of GA. So GID, as I told you, the receptor of gibberellin, full form is gibberellin insensitive dwarf 1, receive the GA, but here as the GA is not there, it will be free from GA. That GID1 will form complex with the with the DELA protein if GA is there, but if it is not there, GID1 will be free, and DELA will now interfere with the transcription factor. This transcription factor is the main component which is responsible for the expression of GA responsive genes, but DELA will block the work of transcription factor. That is why the GA responsive gene will not be expressed. But when GA is present, GA GID complex will be formed first, then this complex will move and it will form GA-GID DELA complex. GA-GID DELA complex will now add with the SCF1 SCF SLY1 complex, which is responsible for the ubiquitination of DELA protein, and that will lead to the degradation of DELA protein. So DELA protein, when degraded, transcription factor will be now free, and that will help in the GA responsive gene expression, and that is how the GA will be expressed. So I think you understood whole the concept of GA signaling, and if you are not getting, you can comment below. Thank you for the patient listening. Thank you.